All right, let's talk second half of central nervous system then. Let's talk here. Now we'll pick up with diencephalon. So the second half of the central nervous system here, then we'll go with diencephalon now. When we talk diencephalon, I'd like you to know that the diencephalon, you can see here, is going to be made up of three paired structures. Now, when we go through and we talk three paired structures, we'll go through and we'll look at each of these paired structures individually and talk about them one by one as we progress. So here when we go through and we talk diencephalon, first I'd like you to know that the diencephalon is going to form the central core. It will form the central core of the forebrain. This diencephalon is surrounded by the cerebral hemispheres. It's surrounded by the two cerebral hemispheres. And again, when we talk diencephalon is going to consist of three paired structures. Those three paired structures, as we can see here, are going to include the thalamus, hypothalamus, and the epithalamus. Now, when we talk about the diencephalon are gray matter areas. They are gray matter areas that enclose the third ventricle. If you recall, when we talked primary brain vesicles, secondary brain vesicles, we saw we had ventricles at the center. So you have to go back and recall that and exactly which ventricle we have there. Now you know is the third ventricle again. So what we'll do is we'll go through and we'll talk first thalamus, and then we'll move to hypothalamus, and then we'll check out epithalamus. So when we talk thalamus, its organization, I want you to know when we talk thalamus, here we can appreciate a nice image depicting to us first all three components of the diencephalon. You've got first right inside of here the thalamus, and then here we've got the hypothalamus, and then right back here is where we're going to have the epithalamus. Now here when we go through and we talk thalamus, so that's what we're looking at here, we'll look at this structure then in greater detail. So here we can see thalamus, 80% of it is a diencephalon, and we're going to see it's going to form the superior lateral walls of the third ventricles, It will form the superior lateral walls of the third ventricle. And when we talk thalamus is described as basically bilateral nuclei. They're bilateral nuclei that are connected by the interthalamic adhesion, as we can see here, or the intermediate mass. Now, when we go through and we talk thalamus you'll see is again now bilateral nuclei. Okay, they're going to contain several nuclei now. Each of these thalami we'll go through and we'll look at. And they're going to be named for their location as well. We'll go through and we'll see that as we progress and we talk about each of these different thalami there as well. Each of the different nuclei inside the thalami. Now, the nuclei, they're going to project and they're going to receive fibers from the cerebral cortex. Okay, so it's important you remember that cerebral cortex and its function that we went through and we talked about in the first half of chapter 15 and the latter half of 13. So in the last lecture of CNS. Now here when we go through and we talk thalamic functions, you'll see, number one, it's the gateway to the cerebral cortex. It's the gateway to the cerebral cortex. We could think of it as basically as a relay station. So you can think of it as a relay station. And when we compare it to a relay station, you can see here what it's going to do is it's going to sort, edit, and relay this ascending input. Okay? And now these impulses from the hypothalamus are going to be for regulation of emotion and visceral function. Now these impulses from the cerebellum and the basal nuclei are going to be basically to help direct motor cortices. Now, impulses from memory or sensory are involved with integration. Impulses for memory or sensory integration as well are going to be basically passing through. So it sorts, edits, and relays all of this ascending input. It's making sure all of this ascending input is making its way to the exact right spots. Also, again, you can see it mediates sensation, motor activities. So it mediates sensation, motor activities. Also, cortical arousal it's involved with. It's involved with learning and memory as well. So functions, a relay station for incoming information that's making its way to the cerebral cortex. It will sort and edit this information, and it mediates sensation, responsible for mediating motor activities, cortical arousal, learning, and memory as well. Here we can appreciate now a nice view of the thalamus in greater detail now. here you can appreciate all the various nuclei within the thalamus. Now, each of these nuclei have their own functional specialties. Now, here I'll give you one. You can see, and I want you to know these in basic idea. They're basically functions. Now, here when we go through and we talk, we can see here again a whole bunch of different nuclei. And here you can see you got the ventral posterior lateral nucleus. This ventral posterior lateral nucleus, it receives impulses from your general somatic sensory receptors, like receptors that are involved with touch, pressure, and pain. So just like that, you can see each of these are going to have their own functional specialties. Next, then, we're going to see we have then what we call the hypothalamus. Now, the hypothalamus, it's named for its position. It's going to form the inferior lateral walls of the third ventricle, and it also will contain many nuclei for which you're going to see, you're going to have to know the functional specialties for it as well. So here, when we go through and we talk hypothalamus, first let's talk organization. It's going to cap the brain stem. It caps the brain stem, and it forms, again, we said the inferior lateral walls of the third ventricle. And the hypothalamus, it extends from the optic chiasma. So here in this view, you can appreciate it extends from the optic chiasma, and it goes all the way to the dorsal aspect, the posterior aspect you can think of the mammillary bodies. So it's making its way all the way from this optic chiasma all the way to that posterior aspect of the mammillary bodies. Again, bunch of nuclei in here as well. Okay, mammillary bodies are examples of one pair of nuclei. And we're going to go through, we're going to see when you talk mammillary bodies, they're paired P-like nuclei. They're paired P-like nuclei. You can see are bulging from the hypothalamus. And they are relay stations in the olfactory pathways. They are relay stations in the olfactory pathway. So just like this, you're going to have to go through and get the functions of each of these various nuclei down. By the time we get to physio, you're going to have every single one of them down. Supraoptic, we're going to see here. By the time we get to endocrine, suprachiasmatic, we're going to see there again. And paraventricular, we'll go through and we'll talk about and we'll check out. So each of these, you're going to have to know basically kind of what their functions are there. Next in here, we're going to go through and we'll talk generally speaking function of the hypothalamus. Now, with all of these different nuclei there, we'll talk generally now the functions here as well. Now, generally speaking, the functions are going to include, number one, you can see it's going to behave as the autonomic control center, our autonomic control center, our autonomic nervous system's control center you can think of. All right, we talked ANS. It's a system of peripheral nerves that are going to regulate cardiac and smooth muscle, and then also the secretions by our glands. Hypothalamus, also involved with the emotional response. It's involved in the perception of pleasure, fear, and rage. So it's involved in the perception of pleasure, fear, and rage, as well as those in biological rhythms, and drives. Body temperature regulation, another function we have here, body temperature regulation. And when we talk body temperature regulation, you'll see hypothalamus is involved with causing sweating for cooling and shivering then to help generate heat. Regulation of food intake, we can see as another function. It regulates the feelings of hunger and satiety. Basically hunger and fullness. Hypothalamus is also involved in the regulation of water balance and thirst. You'll see we have osmoreceptors. And these osmoreceptors, they excite nuclei that trigger ADH's release. They trigger ADH's release from that posterior pituitary. And then ADH, which stands for antidiuretic hormone, also known as vasopressin. ADH causes the kidneys then to retain water, antidiuretic hormone. Now, the same conditions, they stimulate hypothalamic neurons in the thirst center, making us feel thirsty. Therefore, causing us to drink more fluids. Next, we also have another function. It's a regulation of sleep cycles. And that's going to happen via the suprachiasmatic nucleus, which is your biological clock. Again, you've got to know that function there. And what it does, it sets sleep cycle timing in response to daylight and darkness cues that we get from our visual pathways. So you can see how different parts are going to be working together. And then another function here is going to be endocrine system control. Endocrine system control. We'll see this hypothalamus is going to be responsible for releasing hormones, and it releases inhibiting hormones, which you'll have to know about when we get to endocrine. Supraoptic and paraventricular nuclei there are going to be responsible for producing, as we said, ADH, and then oxytocin, another hormone you're going to have to know about. And oxytocin, so know which one is doing what. Which one is producing ADH, which one is releasing and producing oxytocin, all stuff you'll be responsible for knowing about there. And then in greater detail when we get to endocrine system. Next then we've got the epithalamus. Epithalamus, and when we talk epithalamus, okay, so here you've got all the different functions we've gone through and we've mentioned there. Make sure you've got them down in greater detail. You understand what each of those functions there are. So here then we can see now the epithalamus. Now when we talk epithalamus, we can see now is going to be found at the most dorsal portion of the diencephalon. Found at the most dorsal portion of the diencephalon. And it forms the roof of the third ventricle. It forms the roof of the third ventricle. When we talk epithalamus, main gland we're going to see here is going to be the pineal gland, also known as pineal body. Now this pineal body is found extending from the posterior border. It's extending from the posterior border, and you'll see it's visible externally. And it is visible externally. This pineal gland is responsible for secreting a hormone called melatonin. Melatonin. Melatonin comes from serotonin. Melatonin is a sleep-inducing signal. It's a sleep-inducing signal, and it behaves as an antioxidant. So it helps regulate the sleep-wake cycle, and we believe it functions as an antioxidant. Next in here we can move down and we can appreciate again that diencephalon, thalamus, hypothalamus, and then epithalamus right back in here. So when we say it's visible externally, you can see all you have to do is push the cerebellum down gently, pushing it down very gently, and then you're able to appreciate that pineal gland right back inside of there. And if we're in the lab you'd actually have sheep brains, and the sheep brain then you would be able to see, you'd be able to do that, and the whole sheep brain you'd be able to gently push this cerebellum down from the cerebellum basically, push the cerebellum down from the cerebral, from each of these cerebral basically components. I want to say the proper term I want to use now here is from each of the cerebral hemispheres. Okay, so you'd push down basically the cerebellum from each of the cerebral hemispheres basically gently, and you'd be able to see right underneath there, just like you see here, that pineal gland. And then you've got also that corpora quadrigemina you can see, and then between the two, the posterior commissure. Next, and moving down, we've got then the pineal gland. We've taken care of epithalamus, the brainstem. So let's talk then in green. Now we've got brainstem. When we talk brainstem, we're going to go through and we're going to see now, when we talk brainstem is going to be made up of basically a few different parts. So here we can see in green, the brainstem is going to be made up of number one, midbrain, and we're going to go through, we're going to talk pons, and then we'll check out the medulla oblongata. So let's talk brainstem. Brainstem So from top to bottom, you saw the three major components there. It accounts for about 2.5% of the total brain mass. You can see it's not that big. Histologically, brainstem is similar to the spinal cord, where you have deep gray matter surrounded by white matter tracts. We looked at that when we looked at the organization. And the brainstem is going to be responsible for producing, so its centers, they produce rigidly programmed automatic behaviors. They produce rigidly programmed automatic behaviors that are necessary for survival. Now, the nuclei here are associated with 10 of the 12 pairs of cranial nerves you're going to go through and we're going to check out. So first, midbrain. Midbrain is located between the diencephalon and the pons. You can see that there very well, between the diencephalon and the pons. And here you've got first your cerebral peduncles. Your cerebral peduncles are going to be two bulges. They are two bulges that are located on the ventral aspect, and they form vertical pillars. They form vertical pillars. These vertical pillars are going to help hold up the cerebrum. They help hold up the cerebrum. And they contain large pyramidal, or you can say cortical spinal, motor tracts that are descending towards the spinal cord. So again, two bulges located on the ventral aspect. They form vertical pillars that hold up the cerebrum, and they contain large pyramidal motor tracts descending towards the spinal cord. So let's look at that in greater detail. To appreciate that in greater detail, then, here we can move from this slide here over basically here, and you can appreciate now each of those cerebral peduncles. So here they are on that ventral aspect. So flipping it around, looking at the lateral aspect, here you can see those peduncles there now. Now these peduncles, okay, we can also appreciate here superior cerebellar peduncles. Now the superior cerebellar peduncles you can see there are again fiber tracts, and they connect the midbrain to the cerebellum dorsally. Excuse me, they're going to connect the midbrain. They will connect the midbrain to the cerebellum dorsally. Then you can appreciate here as well the corpora quadrigemina. So here you can see two of those components. Here you got all four. Coming back to this view here, you can appreciate that corpora quadrigemina as we talked about before. So we talk corpora quadrigemina. You can see quadrigemina, four components we're going to be checking out here. So there are four dome-like protrusions. There are four dome-like protrusions on the dorsal midbrain. They are nuclei scattered in surrounding white matter of the midbrain. First you have your two superior, and then you have the two inferior. So the two superior are going to be known as the superior colliculi. When we talk superior colliculi, the superior colliculi are going to be visual reflex centers. They are visual reflex centers. And what they do is they coordinate head-eye movements. They coordinate head-eye movements when we visually follow a moving object. So that's the superior colliculi, you can see there very nicely. And then the inferior colliculi now are your auditory relays. Now they're auditory relays from your hearing receptors of the ear to the sensory cortex. So they're going to be auditory relays from hearing receptors of the ear to the sensory cortex. And they are going to act in reflexive response. They will act in reflexive response to sound, such as in the startle reflex, in which you turn your head towards an unexpected sound. So it's your inferior colliculi. Inferior colliculi. Next then we have our periaqueductal gray matter. Now when we talk periaqueductal gray matter, the periaqueductal gray matter, we're going to move over to this picture here. You can appreciate that there very nicely. Now this periaqueductal gray matter, you can see is going to be found surrounding the cerebral aqueduct. It's found surrounding the cerebral aqueduct. which runs through the midbrain. This periaqueductal gray matter is involved in pain suppression. It's involved in defensive behaviors, reproductive behavior, and in consciousness. And it's involved in consciousness. Next up, we've got the substantia nigra. When we talk substantia nigra are going to be pigmented nuclei. Pigmented nuclei that are embedded in each side of the midbrain white matter. Pigmented nuclei embedded in each side of the midbrain white matter. They're located deep to the cerebral peduncles. Located deep to the cerebral peduncles. You can see the substantia nigra have a nice dark color. This nice dark color, it reflects a high content of melanin. It reflects a high content of melanin. Melanin is going to be a precursor of the neurotransmitter dopamine. It's a precursor of the neurotransmitter dopamine that's released by these neurons. So if you have a degeneration of the dopamine-releasing neurons of the substantia nigra, so when you have a degeneration of the dopamine-releasing neurons of the substantia nigra, you'll see this is the ultimate cause of Parkinson's. Deep to the substantia nigra, we can appreciate the red nucleus. The red nucleus are also pigmented nuclei. Embedded in each side of the midbrain's white matter. Embedded into each side of the midbrain's white matter. The red nucleus, you can see, lies deep to the substantia nigra. They lie deep to the substantia nigra. And you can see they've got a nice reddish color. That reddish color is due to its rich blood supply. It's due to its rich blood supply and iron pigment. The red nucleus are relay nuclei. They're relay nuclei in descending motor pathways that are going to affect limb flexion. Next then we've got our pons. We talk pons. Here you can appreciate then the pons, right? Inferior to then that midbrain. So we talk pons. Pons is the bulging brainstem region. It is the bulging brainstem region wedged between the midbrain and the medulla oblongata. Wedged between the midbrain and the medulla oblongata. Dorsally, it forms the anterior wall of the fourth ventricle. Dorsally, it's going to form the anterior wall of the fourth ventricle. And as you can see here in this picture, it's composed of conduction tracts. It is composed of conduction tracts. Now here we can appreciate the middle cerebellar peduncles. Now these middle cerebellar peduncles, they're superficial ventral fibers. They are superficial ventral fibers and they are orientated transversely. And dorsally. So they're orientated transversely and dorsally. And what they're going to do is they are going to connect the pons. They will connect the pons bilaterally with the two sides of the cerebellum dorsally. Next, then we've got the medulla oblongata. Now, the medulla oblongata, also known as just medulla, it forms the most inferior portion or the inferior part of the brainstem. It blends into the spinal cord at the level of the foramen magnum. The central canal of the spinal cord continues upward into the medulla, where it's going to broaden out to form the cavity of the fourth ventricle that you have back there. Here we can appreciate our pyramids. Now the pyramids we'll see are going to be, so we'll move back out to this ventral view. So here you can appreciate pyramids bilaterally. Pyramids are two longitudinal ridges. They are two longitudinal ridges that are flanking the midline on the medulla's ventral aspect. They're formed by the large pyramidal tracts descending from the motor cortex. Here you can also appreciate the decussation of the pyramids. When we talk decussation, it's the crossover point where most of those fibers are going to cross over to the opposite side. Where they cross over to the opposite side and then continue down the spinal cord. So here we would have something like this. These fibers are coming down here, crossing over, and then coming down on that side. Fibers from this side are coming down, crossing over, and making their way down the spinal cord on that side. So you can see why now. This is also helping explain why some things are going to be controlled from one side of the brain on the opposite side of the body and whatnot. Just like we saw with our optic nerve. So here then we can appreciate next, and we're going to see we've got our inferior cerebellar peduncles. Now inferior cerebellar peduncles you can appreciate here. Inferior cerebellar peduncles, fiber tracts again. They're going to connect the medulla to the cerebellum dorsally. They will connect the medulla now to the cerebellum dorsally. Next then we've got the olives and the inferior olivary nuclei. So let's talk olives. Olives are going to be found lateral to the pyramids. So here you can see, if we were to go back to this ventral view, you can't really appreciate them there. You can see them here off on the sides. So what we'll do is we'll turn this whole brainstem around. Turn it around here, you can appreciate now the olives very nicely. So when we talk olives, olives are going to be oval swellings. So lateral to the pyramids, they're oval swellings, which are mainly caused by wavy folds of gray matter. And what they are, they're relaying, what they're doing is basically they're relaying sensory information. They're going to be relaying sensory information on the state of stretch, on the state of the stretch of muscles and our joints to the cerebellum. So it's going to be basically relaying sensory information on the state of stretch of muscles and joints to the cerebellum. Then we've got the vestibular nuclear complex. Now the vestibular nuclear complex, then, we're going to be able to appreciate now by us moving over, and by us moving over to, you can see now, this slide, vestibular nuclear complex. Okay, actually, no, I'm sorry. When we talk vestibular nuclear complex, I want you to be able to basically see it here. Phone call coming in. Try to avoid that and keep us on point. Okay, so here now, when we talk vestibular nuclear complex, I want you to know collectively, now it's going to include basically the vestibular nuclei. So here when we talk function, that's the main thing you need to get from here, is that it's going to be responsible for mediating responses that maintain equilibrium. It mediates responses that maintain equilibrium. So now when we talk function of the medulla, When we talk function of the medulla, you'll see it's going to have various functions here. First function I'd like you to know about is that it's going to contain our cardiovascular center. It contains the cardiovascular center. Now, this cardiovascular center itself is made up of two subcenters. We'll see first you have the cardiac center itself. The cardiac center is going to be responsible for adjusting the force and the rate. It adjusts the force and the rate of heart contraction. Now, vasomotor center you have here as well. The vasomotor center, it changes blood vessel diameter to regulate blood pressure. It will regulate blood vessels, so it changes blood vessel diameter to regulate blood pressure. You also have your respiratory center here. When we talk respiratory center, we'll see the respiratory center is going to be involved with controlling the rate and the depth of breathing. It's involved in controlling the rate and the depth of breathing. There's also other visceral centers here as well. Now, when we talk other visceral centers, these other visceral centers are going to include centers for vomiting, centers for hiccuping, swallowing, coughing, and even sneezing are all going to be found located at the medulla. So it's a very, very important region there, and its functions are very important there as well. Next one, we've got the cerebellum. When we talk cerebellum, cerebellum is also known as a small brain. The cerebellum is cauliflower-like. It is cauliflower-like. It's located dorsal to the pons and the medulla. It's going to be located dorsal to the pons and the medulla. So let's move over and check that out. You can see that there. You can see that there as well. It protrudes under the occipital lobes. It protrudes under the occipital lobes of the cerebral hemispheres. So it protrudes under the occipital lobes of the cerebral hemispheres, from which it's separated as well. From which it's going to be separated. It's going to be separated from the cerebral hemispheres by the transverse cerebral fissure. By the transverse cerebral fissure. So now some structures in relation to the cerebral hemisphere. First, you can appreciate the cerebellar, in relation to the cerebellum, are going to be first the cerebellar hemispheres. Now we talk cerebellar hemispheres, they're apple-sized hemispheres. They're apple-sized, and they're connected medially. They're going to be connected medially to one another. You can see that right inside of here. Where my arrow went. There we go. So here, so they are apple-sized, connected to one another, apple-sized, connected to one another, medially by the vermis, connected to one another medially by the vermis. So when we talk vermis, that's the structure you're seeing right here in the center. We talk vermis. Vermis is this worm-like structure. It's a worm-like structure, and it connects the cerebellar hemispheres, as you can see there. It connects the two cerebellar hemispheres. Now here you can also appreciate the folia. Folia are going to be pleat-like gyri. They are pleat-like gyri, basically, found on the surface of this cerebellum. Now here you can see the cerebellum is going to get divided into an anterior lobe and a posterior lobe. Here you can appreciate that anterior lobe right on top. And then here we can appreciate the posterior lobe. Now when we talk anterior lobe and posterior lobe, you can appreciate both of those there very nicely. They're going to be separated from one another. They will be separated from one another, you can see, thanks to the primary fissure. Thanks to the primary fissure. So the primary fissure separating that anterior lobe from the posterior lobe. And then here we can see in relation to the posterior lobe, we've got the horizontal fissure. Horizontal fissure. So you're running completely horizontal. Next then we've got the flocculonodular lobe. So moving back, here you can appreciate now the flocculonodular lobe. Now the flocculonodular lobe, small propeller-shaped lobe. It's found deep to the vermis. It's deep to the vermis and the posterior lobe. Deep to the vermis and the posterior lobe. It cannot be seen on the surface view. It cannot be seen on the surface view. And what it does, it receives input. It receives input from equilibrium apparatus. It receives input from our equilibrium apparatus. And then it adjusts posture to maintain balance. It will adjust posture to maintain balance. And then here we can appreciate the cortex. When we talk about the cortex I want you to know is this thin outer layer of gray matter. It's this thin outer layer of gray matter. Here we've got also Purkinje cells. They're going to be large cells. They've got extensively branched dendrites. And what they do is they send their axons. They send axons through the white matter to synapse with the central nuclei of the cerebellum. So these Purkinje cells are going to be large cells with branched dendrites. And they send axons through white matter to synapse with the central nuclei of the cerebellum. So you can see the white matter. The white matter, it's got this distinctive pattern. We refer to it as the arbor vitae. So white matter, you can see then running through the center of that cerebellum, takes on this distinctive pattern. And we refer to it then as the arbor vitae. The arbor vitae. Next, there's dentate nuclei, deeply situated paired masses of gray matter. Dentate nuclei there as well, paired masses of gray matter. So I want you to go through and get their functions down as well. Next, let's talk functions of, generally speaking, the cerebellum. So when we talk cerebellum, it's involved with coordination and cognition. So first, coordination, it coordinates body movements. It uses the cerebellar peduncles for cerebellar processing. Cognition, it recognizes and it predicts sequences of events. It's involved with recognizing and predicting sequences of events, so that way we can adjust. And then some non-motor functions. Non-motor functions are going to include, we'll see, word association and puzzle solving. It's involved with word association and puzzle solving. And then you've got the term ipsilateralization there as well. We've talked contralateralization. So ipsilateralization, basically, you've got there as well, talking about functions being same-sided there. Next, we have the functional brain systems. And we talk functional brain systems, networks of neurons. They're going to be networks of neurons that work together, and they span large distances in the brain. So you can't localize them to a specific region. So first we'll talk limbic system. Here we can appreciate limbic system. Limbic system is a group of structures. Now its cerebral structures encircle the upper part of the brainstem. So it's a group of structures. Its cerebral structures encircle the upper part of the brainstem. Now the functions, it's involved with feelings. Basically affective, it's the affective brain. So when you talk functions, it's the affective brain, meaning it's involved with feelings. And emotional reactions. So when you talk location now, location, it's located on the medial aspect of each cerebral hemisphere and diencephalon. Located on the medial aspect of each cerebral hemisphere and diencephalon. One structure I'd like you to know about here is the amygdala. The amygdala you can appreciate here very nicely. When we talk amygdala, the amygdala is going to be an almond-shaped nucleus. It's an almond-shaped nucleus that sits on the tail of the caudate nucleus. And it recognizes facial expressions and elicits fear response. And it elicits fear response. The second structure we have here is the cingulate gyrus. Here we can appreciate the cingulate gyrus. When we talk cingulate gyrus, it's going to play a role in expressing emotions through gestures. It plays a role in expressing emotions through gestures and resolving mental conflicts when frustrated. And resolving mental conflicts when frustrated. Third structure we have here is the hippocampus. Now, the hippocampus, you can appreciate here, it plays a role in memory. It plays a role in memory. And then we have the fornix. Fornix is going to link. It links the limbic system. It's going to link limbic system regions together, along with other fiber tracts. So the fornix links limbic system regions together along with other fiber tracts. Next, then we can appreciate reticular formation. So here when we talk reticular formation is composed of loosely clustered neurons. It's composed of loosely clustered neurons in white matter. In white matter. When we talk functions, the functions of the reticular formation is to send continuous streams of impulses to the cerebral cortex. I mean, the picture is depicting to us that there as well, very well. So it sends continuous streams of impulses to the cerebral cortex. It also filters sensory inputs. And as we can see there, it has a motor arm as well. And it has a motor arm. So the location of reticular formation, it's extending through the central core of the medulla, the pons, and the midbrain. And then go through the structures, go through main nuclei. I want you to know there's raphe nucleus. So go through the raphe nuclei and make sure you got the function down of the raphe nuclei there as well. Next, then, let's talk brain protection. Let's talk brain protection. So when we talk brain protection, here we can see first the biggest significant source of protection is going to be the bone. Above the bone, you got periosteum, and then you got scalp, and above the scalp, then you're going to have hair, at least for the majority of us. Next, then we move down, you've got, you can see now, in here we're going to see meninges. Meninges are going to be 3 connective tissue membranes that are going to lie external to the CNS. And what they do is they cover and they protect the central nervous system. They protect blood vessels. They enclose venous sinuses. They contain CSF. They contain cerebral spinal fluid. And they're going to form partitions. And they form partitions. So from external to internal, we've got first dura mater. The dura mater is leathery. It's the strongest meninx. It surrounds the brain. And what it is it's a 2-layered sheet of fibrous connective tissue. It is a 2-layered sheet of fibrous connective tissue. First layer you have is the periosteal layer. The periosteal layer, as we can see in this picture, is the more superficial layer. It's attached to the inner surface of the skull. It is attached to the inner surface of the skull. The meningeal layer, you can see just deep to that. So the meningeal layer is deeper to the periosteal layer. And the meningeal layer, I want you to know, forms the true external covering of the brain. It forms the true external covering of the brain and continues caudally. And it's going to continue caudally into the vertebral column as the spinal dura mater. As the spinal dura mater. You've got dural sinuses there. They're going to be responsible for collecting venous blood. And they'll direct this blood then to the internal jugular vein. Dural septa we can see here as well. Now, dural septa are going to be flat partitions. Again, you got the term septa there. Flat partitions that are going to subdivide the cranial cavity. There you can think of inward extensions of the meningeal dura mater. So you can appreciate them here in this picture very well. So first here you have the falx cerebri. The falx cerebri you can see here very nicely. It's sickle-shaped, large, sickle-shaped fold that dips into the longitudinal fissure. Between the two cerebral hemispheres. Anteriorly, you can see here it attaches to the crista galli. Next, you have the falx cerebelli. Falx cerebelli is going to be the inferior continuation of the falx cerebri. So it's the inferior continuation, or I'm sorry, the posterior continuation you can think of. The posterior inferior, because it's moving down, of the falx cerebri. And it's going to run along the vermis of the cerebellum. It's going to run along the vermis of the cerebellum. Then you've got the tentorium cerebelli. Tentorium cerebelli, it resembles a tent. It resembles a tent over the cerebellum. So it resembles a tent over the cerebellum. And what it is, it's a horizontal fold. It's a horizontal fold that's going to extend into the transverse fissure. And you can see one half of it there very nicely. It's like a tent you can think of on top of that cerebellum. So that's one side, and then the other side would be proximal to us, which we can't see. So moving back, we've got then our arachnoid matter. Now we talk arachnoid matter. Arachnoid matter, now you can appreciate just deep to the dura mater. It's the middle meninx. It's the middle meninx. It's a loose brain covering. And it's going to be separated from the dura mater thanks to the subdural space. So subdural space is a narrow serous cavity that separates this arachnoid matter from that dura mater. Then you also have a subarachnoid space. The subarachnoid space is found to be a wide space beneath the arachnoid membrane. This layer has, you'll see CSF. And we can appreciate here the arachnoid villi. And they're just basically knob-like projections that protrude through the dura mater and into that superior sagittal sinus. Third layer then is the pia mater. When we talk pia mater, it's a delicate connective tissue, and this pia mater is richly invested with tiny blood vessels. It's richly invested with tiny blood vessels, and this pia mater is going to be found clinging tightly to the brain. It clings tightly to the brain like cellophane wrap. Then we have cerebral spinal fluid, CSF. You'll see the CSF is going to be found in and around the brain and spinal cord. Found in and around the brain and spinal cord. It's a watery broth. When you talk about its functions, you'll see its functions is that it nourishes the brain, number one. It's going to nourish the brain. And also what it does is it provides a liquid cushion. So giving kind of buoyancy you can think of. It gives buoyancy to the CNS structures. Then here we can appreciate the choroid plexus. When we talk choroid plexus, the choroid plexus now will move over, and you can appreciate here the choroid plexuses. The choroid plexuses, they're going to be basically hanging from the roof of each ventricle, as you can see, and they form CSF. They're going to form the cerebral spinal fluid. Those choroid plexuses are going to be enclosed by pia mater, and then a layer of ependymal cells. So when we talk blood-brain barrier, function of the blood-brain barrier is that it's protective. It's a protective mechanism that's going to help maintain a stable environment for the brain. So there, if you recall, we talked about some certain cells. We had astrocytes, and basically the major cell there between that the components of the brain, the neurons, those different cells were helping to connect basically the nutrient supply line to different parts of the brain tissue. So you got the components there. Make sure you got every one of those understood there. and then understand the variation. You'll see, go through and check that out. I want you guys to go through and look at the variation of the blood-brain barrier in the different parts of the brain and the ventricles. Next, then let's move to spinal cord. Let's talk spinal cord. So when we talk spinal cord, here we're going to be able to see the spinal cord. It's about 42 centimeters long, 17 inches long, you can think of. About 3/4 of an inch thick. About 3/4 of an inch thick. The spinal cord is located enclosed in the vertebral column. It's found located enclosed within that vertebral column. And it extends, if you recall, you can see here from that foramen magnum down to the level of the first or second lumbar vertebra. Down to the level of the first or second lumbar vertebra, just inferior to the ribs. So what's the function? It's going to be involved with conduction, neurointegration, locomotion, and reflexes. So make sure you go through each of them and understand each of those functions there very well. It's got protection as well. So we talk protection. First, you've got the vertebral column itself. Then we've got the meninges. Now we talk meninges, we've got spinal dura mater. The spinal dura mater is going to be a single layered matter. It's not attached to the bony walls. And we're also going to be able to appreciate here epidural space. The epidural space is going to contain fat. So it's filled with a soft padding of fat, adipose, and a network of veins. So you can see that here very nicely. This epidural space is between the bony vertebra, and the spinal dura mater. Again, you got to use simple English, like 5th, 6th grade English, right? Prefix epi, sub, okay, and so forth. So epidural space, so between the bony vertebra and the spinal dura mater. Here you can see they're going deeper. They're going to the subarachnoid space. So again, you got to use those prefixes to your advantage. Basic English, like 5th grade English, 4th grade, I think probably. Next in here, we're going to see we've got, after the epidural space, our arachnoid matter and then the pia mater. So arachnoid matter and pia mater, then you can see right from this view here, but again, that view is not doing justice to it. You can see here, dura, arachnoid, and then you've got the pia mater right on top. Next in here, let's move through and let's talk then CSF we've mentioned. Let's talk microscopic structures here. You got first your denticulate ligaments. They're going to be saw-toothed, basically shelves of pia mater. Saw-toothed shelves of pia mater. Got a good picture. Well, you guys can Google that picture there as well. So they're going to be saw-toothed shelves of pia mater. And what they're going to do is they're going to help secure the spinal cord to the tough dura mater. They will help secure the spinal cord to the tough dura mater. Next then is a cervical enlargement. Now when we talk cervical enlargement is the region where the spinal cord is going to be about the width of the thumb. So we can come over to this picture here. I'm sorry, so let's come back here. So cervical enlargement is going to be a region where the spinal cord is going to be about the width of a thumb. About the width of your thumb. So here you can see, right in this area here is where that kind of enlarges and it kind of thins out. And then we move down, you get to the cervical enlargement. Same thing, it kind of thickens up right in that area there as well. Moving down then, we've got the medullary cone or conus medullaris. Now the medullary cone or conus medullaris is going to be where the cord terminates. It's where the cord terminates. It's found at the inferior aspect. It's a tapering cone-shaped spinal cord terminating area. Tapering cone-shaped spinal cord terminating area. Then we can appreciate the cauda equina. when we talk cauda equina, the cauda equina is going to be a collection of nerve roots. It's a collection of nerve roots at the inferior end of the vertebral column. Collection of nerve roots at the inferior end you can think of the spinal cord or vertebral column. So right inside of here, like a horse tail. Cauda, caudal, equina, horse. Phylum terminale you can appreciate in here as well, the terminal phylum. Phylum terminale. It's a fibrous extension of that conus medullaris. It's a fibrous extension of the conus medullaris. It's covered by pia mater. It's covered by pia mater. And it extends inferiorly. It extends inferiorly from the conus medullaris down to the coccyx. You can see that there very nicely. Down to the coccyx to help secure and anchor that spinal cord in place. Helps to secure or anchor the spinal cord in place. Now some microscopic structures we're going to be able to appreciate here. When we talk microscopic structures, first we can appreciate here, you can see the anterior or ventral median fissure and then the dorsal or posterior median sulcus. So here you've got the dorsal or posterior median sulcus and then anterior or ventral median fissure. Basically, they're running the length of the cord and dividing the cord in halves. They run the length of the cord and divide the cord in halves. You can appreciate here now gray matter. The gray matter in this cross section, it looks like a butterfly or the letter H. Here you've got first gray commissure. Gray commissure is a crossbar of gray matter that encloses the central canal. You can see that there very nicely. And then you can see the dorsal horns of gray matter. The dorsal horns of gray matter are dorsal projections of gray matter. Dorsal projections of gray matter. Then you've got your dorsal roots coming from the basically rootlets. Dorsal roots, they're formed by your afferent fibers. They're formed by your afferent fibers that are going to be carrying impulses from peripheral sensory receptors and bringing them, basically bringing in these impulses to the CNS. And then your ventral horns you can appreciate here. The ventral horns are going to be ventral projections of gray matter. Ventral projections of gray matter. And here we can appreciate the ventral roots, also derived from the rootlets. The ventral roots now are small. And they're going to be basically motor neurons that send out their axons to skeletal muscle. So we talk ventral roots, motor neurons that send out their axons to skeletal muscle via these rootlets. And then you can see there the lateral horns. Lateral horns, they're going to be a pair of gray matter present in the thoracic and superior lumbar segments only. Present in the thoracic and superior lumbar segments only. And the white matter you can see surrounding that gray matter. And the white matter is basically myelinated and unmyelinated nerve fibers. And it allows communication between the brain and spinal cord. The white matter gets divided up as funiculi. So here we've got first dorsal funiculi, lateral funiculi, and then we can appreciate ventral funiculi or anterior funiculi. So anterior, ventral, lateral, dorsal or posterior funiculi. And then you need to know you've got various types of fibers. Some of your fibers are going to be, again, ascending, making their way up. Some are going to be descending, making their way down, while others are going to be transverse fibers. So that will conclude then the second half of central nervous system. Here we can see we've got then this picture depicting to us basically patient having a needle making their way into the subarachnoid space. So it's a lumbar puncture, basically so they can remove some CSF, cerebrospinal fluid. In situations like meningitis, this can occur. Very nice picture of ganglia. So we've already talked about ganglia. You can see them there in way greater detail now. So no excuse of not knowing what ganglia are after this.